Hello, I'm Andrew, a junior bio student at Hendrix College, and I spent this summer in the salt marsh. So salt marshes are super important ecosystems. They provide habitat for fish, crabs, and shrimp. They prevent the erosion of shorelines, and they also sequester carbon from the atmosphere. However, fishery species, a recent study came out that fishery species are rarely using much of this flooded marsh habitat. They're only going but a few meters into the flooded marsh, and it's really strange because all of this, flooded marsh is super good and seemingly optimal habitat for all these fish. And we think that the reason is because of dissolved oxygen. So our main objective for this study was to determine if dissolved oxygen, or DO as I'll call it, limits more extensive use of the salt marsh. And we hypothesized that the DO is dropping to low or even hypoxic levels, which is driving out these fish out into the more oxygenated open water. So to test this, I've got a little DO logger. I'll normally give this to you. So this is a hobo DO logger, and we place this at 10, 5, and 1 meters into the flooded marsh, and then 1, 5, and 10 meters out into the open water. This gave us a good look at that entire gradient of dissolved oxygen along that marsh edge. I'll set that down. So we looked at 10 sites, and this is listed on this left axis here, and you can also see them on the map. And each site has 6 loggers at those checkpoints, and the loggers are kind of represented by these pie charts. And it looks a little overwhelming, but the pie charts are pretty simple. So each single pie chart, again, represents 1 logger and the amount of time it spent in the salt marsh, which is 48 or more hours, 48 being like the least it could be out there. And the red represents the proportion of time that logger was in less than 2 milligrams per liter DO, which is considered hypoxic. The orange is between 2 and 3, the yellow is between 3 and 4, and then the blue is 4 and up. And if you just look at these 3 in the flooded marsh loggers, you'll see a greater proportion of that red, orange, and yellow, which kind of just shows you how much less dissolved oxygen there is in these locations. And the proportion of blue at these open water sites is much more relatively, and that would mean that there's plenty of oxygen in these sites. These are my time series. This is basically just 1 site's worth of loggers, and they show a very similar pattern. So if you look at this red line, that is our -10 logger, our 10 meters into the marsh. Orange is our -5, yellows are -1, and then the blues are our open water loggers. And if you look at this left axis at dissolved oxygen, you'll see that that red logger is much lower in concentration, and that orange and that yellow are much lower than that blue open water site. You can see the same at that LBP2 site, which is down here. They're both at the Little Bay Peninsula. And you can just see that red, orange, and yellow diving down into these low dissolved oxygen concentrations, and that blue sitting right up top so there's just plenty, relatively, more oxygen. And these findings basically conclude that the flooded marsh is way less concentrated in DO, which makes sense why they wouldn't be living in there or being driven out, because they are seeking that more oxygenated water in the open water. It's just so stressful in that dissolved oxygen, that low dissolved oxygen environment. And our next steps will just be doing more DO logging. We're also going to be looking at the specific tolerances of each fish, kind of find their upper limit of dissolved oxygen, seeing if they would be driven out by these low dissolved oxygen levels. And then we're also going to be looking at in fauna results. They just take a while to process. But yeah, thank you.